My name is Melo. I'm a researcher at the IDPs, the Institute for Bioengineering Biosciences at the University of Lisbon, Instituto Superior Técnico in Portugal. And today I will discuss with you the application of FRET, that is a Förster Resonance Energy Transfer, particularly at the single molecule level, in order to characterize the conformational ensemble of interstitial disorder proteins, so the IDPs. So here I just put an overview of the outline of my training section today, the main aim. So I will start by overview the main principles of FRET and the three main conditions required in order to have FRET between 2 404s. Then I will discuss the advantage, and particularly for IDPs to reach a single molecule level in FRET, in which actually you can characterize heterogeneous systems and determine different conformational states compared with ensemble FRET measurements. Then I also will present some of typical setups to perform this kind of single molecule FRET experiments, particular turf that you don't use too much for IDPs that requires surface immobilization of our molecule of interest, and also confocal, in which we actually study free diffusing species, and that has been mostly used for IDPs. Then I will finish this first part of the training section by also explain how you obtain this double label protein with a donor and a receptor 404 to perform these single molecule FRET experiments. And the second part of my training section, I will discuss also the application of IDP, the single molecule FRET to study IDPs, how we interpret these single molecule FRET data, and in particular for IDPs that you cannot use the Förster equation, the master Förster equation, since I was only to determine a single donor receptor distance. Has for IDPs, you have a distribution of distance. In this case, you use, as a simplest way, polymer physical models in order to convert this mean FRET efficiency to root mean squared end-to-end distance. And finally, I will present a case study based on my previous postdoc work at the lab of Professor Elizabeth Rhodes, in which I focus on tau protein. In this case, I use single molecule FRET to determine the conformational ensemble of tau in complex with solvotubulin, is a biological partner, and also to contrast, for example, when binds tau to heparin, that is aggregation inducer, and show how powerful this method is to actually characterize aggregation-prone proteins, in this case, and the functional states. So I will just move here my pointer to be easier than for the training. So to start and to just briefly review, FRET is a non-additive energy transfer between 2 404s, a donor and a receptor, that happens due to a dipole-dipole coupling mechanism. So the idea, particularly on using intramolecular FRET, is that you can determine distance, intramolecular distance in your biomolecules. As exemplified here, for example, if you label with your protein with a donor and the receptor site specifically, and if this distance is too far apart, this means if you excite your donor, you just record the fluorescence intensity of your donor, and you don't have the contribution of the receptor. But then the conditions that you have FRET in these donor and receptor 404s are in really close vicinity. If you excite in this case, the donor will record the fluorescence intensity of the donor, and the receptor 404 could quantify and determine this FRET efficiency. So the primary conditions to have FRET, first of all, is to have an overlap between the fluorescence emission spectrum, shown here, of your donor, and also the absorption of a spectrum of your receptor. So also the donor and the receptor transition dipoles orientation should not be perpendicular, otherwise you don't have FRET. And also important for us, For this, we use Fred as a spectroscopic ruler. The donor receptor molecules must be in close proximity and below 10 nanometers. And as shown here, this constant of Fred transfer of energy depends on inverse of the 6th power of the distance between these two force. For this, we use Fred as a spectroscopic ruler, as shown here, have this sharp transition of this efficiency of Fred with the distance between these two forces. So the idea behind is that if they are close, so this distance between the receptor and the donor is too low, this means that you have a high Fred efficiency, but if it gets close, not so close and far apart, right? It's a distance start to increase too much and higher even than 10 nanometers, the Fred efficiency is almost 0. So the simplest way to perform these measurements, and I would say in a simple setup, is just to do intensity-based measurements, where we excite, in this case, your donor site specifically, and you record in real time the emission of your donor and the receptor. Of course, there is more complex setups I will not cover here today, but with pulse laser can also record fluorescence lifetime. And so basically, you determine this Fred efficiency by the number of photons record for the receptor as a ratio in between the receptor and the donor, and you perform a rheosymmetric-based measurement as shown here. So basically, again, if this distance is too low, so they are close, this means that the Fred efficiency is high, so the intensity on a receptor channel should be real high compared with a donor. If they are close to the Förster radius that is specific for a specific donor receptor pair, you can calculate the theoretical value. So you'd expect, in this case, the Fred efficiency 50%. So the intensity should be roughly the same in the two channels. And if they are far apart, so the Fred efficiency is really low. This means that intensity of the donor channel is really high. Of course, it's more complex because you always need to correct for detection efficiency of the both detectors, also the quantum fields, but basically it's a rheosymmetric measurement. And so basically record over time in the both channels as shown here. So the Fred efficiency, for example, if you start is really low, this means that if they are far apart, the intensity on the donor channel is high, and the receptor is really low. But at the same time, if then the Fred efficiency should increase, what you should record basically is a drop of intensity of the donor channel with a concomitant increase of intensity on a receptor channel. So you record this over time. So I also like, first of all, to state here that to perform these conformational studies is really important that you perform Fred at the single molecule level. I'm not just saying about IDPs, but in general, because on a sample or bulk measurement, for example, if you have different conformations, and if we just perform a simple experiment, steady state, and record the fluorescence emission spread of both donor and the receptor, basically have a contribution of all species that you have, all conformations. And then at the end, you determine your mean or average Fred value, just a single value. If you go at the single molecule level, this is one of the advantage. Of course, if you use a third setup, you can follow in real time the transition between different states. And if you use, for example, confocal setup, you can have a distribution. This is a histogram of distributions of Fred efficiencies. So what this means, for example, for this case, is that you have 3 different states. So you have a more open conformation with high distance between the donor receptor fluorophore with a low Fred efficiency, kind of one intermediate Fred efficiency state, and then you have a really more compact conformation with a high Fred efficiency. So if you go on ensemble and ensemble average measurement, you just get a single value, but not translate what is meaning your system in these different solutions. And so the way that I usually try to sell this method is that it's a single molecule technique. So this means that you can study heterogeneous system, dynamic process, and also follow conformational change on this nanometer scale. So of course, has been used not just to follow the folding of IDPs, but also global proteins or even nucleic acid proteins and different biological systems, even to follow real time, for example, enzymatic reactions, also molecular motors, and even membrane proteins. So it's a really powerful technique, not just for the field of IDPs. And again, you can really sort different superpopulations. So one of the things that you should always think in mind when you're playing a single molecule FRET experiment is that you need to produce your protein that is double labeled with a donor and acceptor. And you should, first of all, to choose the right fluorophores for performing these measurements. So ideal fluorophores in general for a single molecule method is that it needs to be photostable, to not have glitching during your experiments. Also high brightness. This means a high excision coefficient and quantum yield. Also low intensity fluctuations, because you want to record change on a fluorescence intensity just due to FRET and not other photophysical process. It should be relatively small. Also, of course, have some linker. Should not, itself, the fluorophore should not interact, of course, with your protein that you are studying, but should not introduce any effect in terms of function and also overall conformation. So you need a lot of course, of controls when you are preparing your protein. And also, particularly for choosing an ideal donor receptor fluorophore, you need to have a high, again, overlap between the donor emission and the receptor absorption to have a high Förster radius. Also, the emission of both donor and receptor should not overlap too much, because otherwise you need a lot of corrections. And also similar quantum yields between these donor and receptor fluorophores. So you need basically to produce this double label protein with the acceptor and the donor and be site-specifically labeled, because you need to know where the place, the residues that you are labeled where you put the labeling. So far, the most common strategies is just to make sure that you have two cysteines in a place that you want to label. Of course, requires some protein engineering. So the idea is not to have too much cysteine on your protein, otherwise you have to remove if you are not probing these locations. You can, for example, mutate a natural cysteine to serine. Of course, you need, again, a lot of controls to make sure that it doesn't interfere with your function. And then we engineer to just place two cysteines. And you can, in this case, label with melamide dyes that are specific for TL groups in the cysteines. And so far, we have been using these FRET pairs, Alexa 488 and 594, that is also well parameterized, for example, if you want then complement our experiments with simulation. So there is other strategies, as for example, incorporation of a non-natural amino acid, and then label with quick chemistry. We use more advanced approach as a chemical ligation orienting-based approach. So again, it's really important, the fluorophores that you choose should be small, have a high brightness and photostability. Of course, you cannot use fluorescence proteins, because you have to always take in mind what you imagine, FRET, is the distance between the two fluorophores. It should be small as possible. And also because they are not so photostable and bright, and also because you can only put these fluorescence proteins and see a termini of your protein. And of course, it's not the way that you use to do single molecule FRET. So if you look on the literature, most of the results has been performed in vitro, these experiments. So there is more recent, some work, particularly from Ben Schuller lab, that has performed in-cell single molecule FRET. In this case, requires the expression combinantly bacteria and then double label. And for example, this lab has been introducing by microinjection with small amounts of this double label protein mammalian cells, and then perform these measurements in cell. But so far, again, most of the experiment has been performed in vitro. So one of the challenges to perform single molecule FRET is that you need to have a small elimination volume in order to be detect one molecule at a time. And for this has been actually used two experimental setups. So one is a turf in which actually we mobilize our protein, in this case of interest, goes to the surface. I will not enter too much into detail on this because I haven't used, and it actually has not been used so far for intrinsically disordered proteins. In this case, we can look for this change of fluorescence intensity, both channels for one molecule over time to look for the transition between different conformational states. And we also have the confocal setup in which the volume of illumination around 1 centiliter, in which we don't require mobilization, just look for free diffusing species. And in this case, what you get is these histograms of distribution of Fret efficiency that give information about multiple conformational states as shown here. So the idea behind the confocal setup is that you focus your laser beam through the dichroic mirror and pass through a high numerical aperture objective into your sample. Then you collect your fluorescence emission, then pass for this dichroic, those removal excitation light, then you have a pinhole, and of course at the end you need to split the two signals, the donor and the acceptor, and for this you use also detectors APDs with high sensitivity. So to have an idea is that if you have like one single molecule and goes and diffuse your small confocal volume, so each of the time, actually most of the time you don't have any molecule there inside. As the molecule enters, you have this burst of photons. So the idea is that for each of these bursts, for example, shown here, I calculate the Fret efficiency for this one, then I go other, and I keep going for all the bursts, and then in the end, you have these typical single molecule Fret histograms that have the distribution of the number of events as a function of this Fret efficiency. So this is a kind of a simple histogram that you just have, basically two peaks, right? So you have one here that have a Fret efficiency close to 0. So in most of the case, remember, you have been using so far the labeling at 2 cysteines. So you have an heterogeneous sample in terms of labeling. Most of this peak comes from the donor-only labeled protein. Can have, for example, a donor position 1, just 1, or 2, or just be double labeled with just the donor. As you don't have here the acceptor, of course, the Fret efficiency is 0. Or can be also the donor acceptor labeled protein, but with the Fret efficiency close to 0. So they are far apart. So if you just do as a simple way I explained at the beginning, just excite the donor, you cannot distinguish these ones. For this, you need more complex setup that you also like alternate excite the donor and the acceptor as doing like pi and pulse lasers, but I will not enter too much in detail on this training section. And then this is the second peak comes from the donor acceptor label protein with a high Fret efficiency. And so even if you just do a qualitative measurement, it's important to look for change, for example, of this mean Fret efficiency as the product, for example, binds to other biological partner. Of course, if its Fret efficiency is too high, it indicates it might have a long range of contacts or more a compact conformation. For example, if upon bind it gets more expanded, the Fret efficiency should decrease. So in a more quantitative way to determine distance for particularly intrinsically disordered proteins is more difficult to interpret the data because you cannot use simply the Förster equation directly because you don't have a single donor accepted distance. So these are IDPs, have a high dynamic and flexibility. And particularly if you consider that the fluorophores reorient rapidly is compared with the donor fluorescence lifetime, basically you can state that this is the integral of your basically the Förster equation, and you have this function of distribution of distance. And the simple way to interpret this distribution is use this the polymer physical models has been used so far. In most of the case, the Gaussian chain polymer model, also the worm-like chain, the self-avoidant walk, but most of the case actually has been using as a simple approximation the Gaussian chain polymer model, where you can actually determine the mean end-to-end distance. Just now to finish, I will present a case study of a previous work that I performed during my postdoc at the level of Professor Elizabeth Rhodes at UPenn at Yale, which I focus on tau. So tau is an intrinsic disorder protein that is associated with several neurodegenerative disease, where the most well-known is Alzheimer's disease. And the main aim of this project was to determine the conformational ensemble in complex with tubulin. To just overview, tau is an IDP that binds to microtubules, also stabilize microtubules. And on this neurodegenerative disorder, it's been proposed that there is this pathological aggregation of rectangles, and that also the loss of function of tau, this means interaction with microtubules, can also contribute for this pathology. So it's really important to understand, for my point of view, for these aggregation proteins, how they aggregate, but how they actually also lost the function. In particular, for tau, you know the main biological function, and I would say for decades, most of the work just focused on interaction with microtubules or how tau actually polymerizes this tubulin into microtubules. And in this work, I focus on this first step of microtubule polymerization, the interaction with tubulin that is basically the building blocks of microtubules. But in order to capture this interaction, you need to go to a single molecule level, because tau decreases the critical concentration for tubulin polymerization. Even a small amount of tau will immediately shift this enclave towards this direction. So in order to look for unpolymerized tubulin, you need to go to a single molecule level. And particularly tau is a large IDP, is also difficult to get conformational information by more standard techniques as NMR. So I will not present all results today. I will just bring some of, I think it's the most interesting ones, and to illustrate how you could find our single molecule FRET experiment. So I here today, I will present just for the longest size of form of tau is a 2N4R, as shown here, contains three main domains: the projection domain, that, as the name indicates, projects away from microtubules. Then you have the problem-enriched region, and also the microtubule binding region, the most important domain in tau, that actually direct interface with microtubules and also tubulin. So as you see, it's a long intrinsic disorder protein. So for this, I create multiple single molecule FRET constructs, because the idea is that it doesn't make sense to just determine one distance in order to get the structural information. So you need to always to have an idea, it's a topology of the protein conformational ensemble to create multiple constructs. And you always, basically, you always have express and also have overland protein, and you just change the place where you put the donor and the receptor for labeling, right? So these are not fragments of the protein. You are always measure FRET in the context of the overland protein. So I always start like designing a construct to probe almost all dimensions of the protein. For this case, I put it at position 17 and also for 33. Then one is to look for long-range contacts between this N-terminus and the microtubule binding region, and this last between the C-terminus and the microtubule binding region. And then I probe the domain, the more functional domain of tau, the microtubule binding region. So for example, this first one spends almost all this domain, and the other to look for the distance between each of the repeats. So if you look, you should keep also, it's really important, keep some of the landing positions in place and just change the ladder, because you need to create some kind of triangulations to then have an idea how P in 3D. So I not have time, of course, to present all data here, but the idea is that you always measure in this, for this work, the FRET for tau just in solution, and when it's fully bound to tubulin, using 10 micromolar, that you know for other complementary FCS measurements, in particular for acid calculation spectroscopy that you use, that tau is bound to tubulin under this condition is fully bound, and also is able to bind to multiple dimers. So as shown here for in gray histograms, you have the one obtained for just the protein in solution, and it will when you have fully bound tau to tubulin and micromolar. And here on the top, I have a cartoon how it looks like tau bound to tubulin and where you have the landing position, just to be easily to identify. So again, we start by this construct that spans almost all this protein, you see that the fat efficiency will be high compared with the theoretical random coil protein, would expect to be 0. What this means that you have these long range contacts between N and C termini. But more important, when you have tubulin, you see that the fat efficiency shifts almost open up, of course, our microscopes to almost everyone. And thank you for the attention.
Thank you, Anna, for the training session. It was very, very interesting. I'm going to stop the recording.